The nervous system. The functional divisions are sensory afferent to CNS and motor afferent from CNS. Divisions by location: visceral, sympathetic, parasympathetic, somatic. Visceral internal organs: deep, ANS motor; somatic, superficial, skeletal muscles, bones, joints, skin, and it's motor. Astrocytes are the most abundant CNS neuroglia. They hold neurons in place, physical support the blood and brain barrier. They guide neuron movement. Neuroglia. Microglial cells are defensive cells in the CNS, macrophages. Epidermal cells line cerebrospinal fluid-filled cavities, make and circulate CSF, allow brain to float. Oligodendrocytes have processes that form myelin sheaths around CNS nerve fibers. Neuroglia. Satellite cells and Schwann cells, which form myelin, surround neurons in the PNS. Hold neurons, protect from toxic stuff in blood. Versatile properties of neurons: excitability, irritability, conductivity, secretion, a chemical neurotransmitter. Chromatophilic substance: rough endoplasmic reticulum. Axon hillock: every action potential starts there. Anterograde: away from the body. Retrograde is towards the body. And there's viruses, and in anterograde it's neurotransmitters. Sensory: transmit impulses from sensory receptors towards CNS. It's also unipolar. Motor: carries out impulses from CNS to effectors, multipolar. And interneurons process info. Graded potentials: short-lived. Localized changes in the membrane potential. The stronger the stimulus, the more voltage changes and further current flows. Triggered by stimulus that opens gated ion channels. Results in depolarization or sometimes hyperpolarization. Postsynaptic potential is the result of a neurotransmitter. Postsynaptic potential: excitatory depolarization and inhibitory hyperpolarization. Depolarization of graded potential: the spread and decay of a graded potential. Stimulus, the neurotransmitter, touch, chemicals, light. Depolarization of graded potential: depolarization spreads. Action potentials: principal way neurons send signals and means of long-distance neural communication. Action potentials do not decay over distance as graded potentials do. The events of an action potential. Step 1, the resting state. All gated sodium and potassium channels are closed. The events of the action potential. Depolarization. Sodium channels open, allowing sodium entry. The events of the action potential at plus 30 MV. Repolarization. Sodium channels are closed, potassium channels open, allowing potassium to exit. Hyperpolarization. Some K+ channels remain open and Na channels reset. Slow to close. And it goes from hyperpolarization back to resting to ions move towards that. Propagation of an action potential. Time of propagation of an action potential. It goes from resting at 0 to time to second. It goes to 2 milliseconds. The voltage at 2 milliseconds is plus 30 M, and Na plus goes in. And then the last one is one way only and sensitive to Stimuli, time is 4 milliseconds. Voltage, after it goes up, when it goes down, it ends up at 4 milliseconds. Degree of myelination. Two types of conduction depend on presence or absence of myelin. In bare plasma, membrane voltage decays. In non-myelinated axons, conduction is slow. Conduction is slow because it takes more time for ions for gated channels to move, and this must occur before voltage can be regenerated. Conduction velocity. Group A fibers are the largest diameter, transmit at 150 milliseconds or 300 miles per hour. Myelinated somatic, sensory, and motor fibers of skeletal muscle and joints. B and C groups include A and S, visceral, motor, and sensory fibers that serve visceral organs. Viscera is slow. B and C fibers are the smallest in diameter. B is the intermediate diameter. Those fibers are just slow. The axon of the presynaptic neuron releases neurotransmitters. Cell body of soma of the postsynaptic neuron receives and responds to neurotransmitters. Chemical synapses are slow, allow for information processing and making decisions, and they also allow for greater range of response. Electrical synapses in between heart muscle cells are joined by gap junctions that connect cytoplasm. They're fast but have no ability to process info and make decisions, and communication is very rapid and may be unidirectional or bidirectional. It's all or nothing. Effect on the postsynaptic cell. Excitatory postsynaptic potential. And ESP is a local graded depolarization, and it gets sodium in and potassium out. Inhibitory postsynaptic potential. IPSP is a local graded hyperpolarization. Neurotransmitter binding opens chemically gated ion channel permeable to either K plus or CO1, potassium chloride, and it needs a stronger stimulus to hit the threshold. In the neural integration of EPSPs and IPSPs, no summation. Two stimuli separate in time because EPSPs that do not add together. B, temporal summation. Two excitatory stimuli close in that time, causing EPSPs that add together. C, spatial summation. Two simultaneous stimuli at different locations cause EPSPs to add together. And the last one is spatial summation of EPSPs and IPSPs. Changes in the membrane potential can cancel each other out. Real-time effects of stimuli. Synaptic potential. Synapses that are used frequently respond the quickest. Long-term potential, important for learning and memory. Acetylcholine is the first identified and best understood. Released at neuromuscular junctions, also used by many ANS neurons and some CNS neurons. Synthesized from acetic acid and choline by enzyme choline acetyltransferase. Degraded by enzyme acetylcholine. Neurotransmitters are dopamine, norepinephrine, and epinephrine. And dolamines are serotonin and histamine. The neurotransmitters are made from amino acid tyrosine. Serotonin is made from tryptophan. Histamine is made from the amino acid histidine. GABA is usually inhibitory. Amino acids make up all proteins, therefore it is difficult to prove which are neurotransmitters. Peptides are proteins that act as neurotransmitters. Endorphins reduce pain reception. Gut-brain peptides are somatostatin and cholecystokinin play a role in regulating digestion. Effects determined by the receptor. Excitatory versus inhibitory. Acetylcholine and NE bind to at least two receptor types with opposite effects. A simple reflex arc. So first, is the stimulus, then it goes to the receptor, then sensory neuron, then integration center, then motor neuron, then effector, and then that causes the response.